Welcome to Unit 1, Introduction to Analytical Chemistry. This video covers a key course for the BSc, I Semester 2, DSc 4B Chemistry Paper IV. Our unit covers several key concepts, including an introduction to analytical chemistry, the importance of analysis, analytical processes, and methods of analysis. We will also explore sampling, different types of errors, methods of expressing accuracy, and important statistical concepts like significant figures, mean, median, and standard deviation. Analytical chemistry focuses on obtaining information about the chemical composition, structure, and behavior of matter. It's a vital and diverse field with applications in environmental monitoring, food safety, forensic science, and pharmaceutical development. It provides accurate and precise information on the composition and properties of substances, helping to solve real-world problems, from water contamination to drug development. The main branches of analytical chemistry include quantitative and qualitative analysis, instrumental analysis, bioanalytical, environmental, pharmaceutical, and food analysis. Common analytical techniques used to identify and quantify chemical species include spectroscopy, chromatography, mass spectrometry, electroanalytical techniques, titration, NMR, and X-ray spectroscopy. Analytical chemistry has broad applications. In the pharmaceutical industry, it helps develop new drugs and ensures their safety. Environmental monitoring relies on it to check the quality of air, water, and soil. It's crucial for food safety, forensic science, and clinical chemistry for disease diagnosis and treatment monitoring. Analysis is categorized into two types. Qualitative analysis answers what is present by identifying elements or compounds. Quantitative analysis answers how much is present by determining the amount of the analyte. Often, both types of information are required, such as knowing both the identity and concentration of water pollutants. Methods of analysis can be broadly classified into classical and instrumental methods. Classical methods include gravimetric and volumetric analysis. Instrumental methods encompass optical and electroanalytical techniques. The choice of method depends on the nature of the analyte and the sample matrix. Sampling is essential for analysis, ensuring the extracted small portion truly represents the entire bulk material. For heterogeneous bulk materials, increments are taken randomly to form a gross sample. This is then divided into a subsample, which, after treatment like size reduction or mixing, becomes the analytical sample. For gas sampling, glass devices with stopcocks are used, and flushing removes contamination. Homogeneity issues can arise with temperature or pressure changes. For liquids, stirring is usually sufficient for pure samples. Heterogeneous liquids require samples from various depths and locations. Sampling of solids is challenging due to potential non-homogeneity and localized impurities. Particle size is a crucial factor. Large samples are reduced to a uniform particle size using crushers and mills, and then the sample mass is reduced. The principle is to systematically select and combine numerous portions. Analysts aim for results as close to the true value as possible. Differences between successive measurements are common. The difference between the average value and the true value determines the result's reliability. These differences are defined as errors, which is the actual difference between the true and experimental result. Errors in quantitative analysis are classified as determinate or systematic and indeterminate or random. Determinate errors can be avoided, their magnitude determined, and they affect all observations in a series similarly. They include personal, instrumental/reagent, methodic, operational, and additive or proportional errors. Personal errors arise from the individual analyst's inability to make accurate observations, such as errors in judging color changes during titrations, reading a burette, or mechanical loss of material. Instrumental or reagent errors result from faulty construction, uncalibrated weights or glassware, or the use of impure reagents. Methodic errors are serious because they are often hard to detect. Examples include wrongly standardized pH meters or background absorption in spectroscopy. Operational errors are physical in nature and not connected with the method itself, often occurring when analytical guidelines are not followed, such as incomplete sample drying or incorrect solution transfer techniques. Accuracy is the closeness of a measured value to the true value, achieved by taking small readings to reduce calculation error. Precision is the closeness of multiple measurements to each other. A set of precise measurements may not necessarily be accurate, as precision is independent of accuracy. This chart visually represents the relationship between accuracy and precision. Results can be highly accurate and precise but inaccurate. accurate but imprecise, or both inaccurate and imprecise. Accuracy can be expressed using absolute error and relative error. Absolute error is the difference between the observed and true value. Relative error is the absolute error divided by the true value and measures the accuracy of the measurement.
Relative error is typically expressed as a percentage, or in parts per thousand. Relative error in percentage is the relative error multiplied by 100, and in parts per thousand, it is multiplied by 1,000. Examples are provided to illustrate the calculation of absolute and relative errors. Significant figures are the digits that carry meaning towards the resolution of a measurement, accounting for inherent uncertainty. All non-zero digits are significant. Zeros between non-zero digits are significant. A trailing or final zero in the decimal portion is also significant. Key rules for significant figures include: non-zero digits are always significant. Leading zeros before a non-zero digit are not significant. Zeros between two non-zero digits are significant. Zeros at the end of a number after a decimal point are significant. The mean represents the average of a data collection. It is calculated by summing all values and dividing by the total number of values. It is a fundamental concept in data analysis, as illustrated by the example calculation for chlorine determination results. Median is the middle value of a replicate set of results, about which all other values are equally distributed. When observations are arranged in order, the median divides the array into two equal parts. The mean and median may or may not be the same. This slide is for visual content related to mean and median, perhaps showing a numerical example or diagram. Mean deviation measures the average deviation of values from the mean of the dataset. It is calculated based on the absolute differences between each data point and the mean. This slide is likely for visual content or further explanation related to mean deviation. Standard deviation is a measure of the amount of variation or dispersion of a set of values. A low standard deviation indicates that the data points tend to be close to the mean, while a high standard deviation indicates that the data points are spread out over a wider range. This slide concludes our introductory unit on analytical chemistry, providing space for any final thoughts or a summary of the statistical methods covered.